Here we go. We're going to do section 8.7, the last section in this chapter. That's going to be about mutations. Remember to have your power notes handy and ready. And also, if you look on Classroom where you found this video, I don't know if you noticed, but there was a word bank or like a sentence bank at the top. Those are all the terms or words that will go into the power notes. So you can kind of select from there, and I hope that helps make the notes more clear for you. So make sure you have that in front of you, your piece of paper, the power notes paper, and the classroom word bank. All right, so mutations. Mutations are changes in DNA. They may or may not affect the phenotype. Phenotype, just think of the pH, and phenotype, think of physical appearance. So that is, they may or may not physically change somebody, change the way their heart works, or their height or something. They may just be something that happens and has no negative effect. So some mutations affect a single gene while others affect an entire chromosome. So remember, a gene, you could think of like the projects, the DNA projects you guys did, like that little piece of DNA. That's too small, but you could think of that as being like a gene, like a certain number of DNA molecules together making a gene. Whereas a chromosome, we're talking about, just thousands and thousands of DNA molecules, hundreds of genes. All right, so chromosome is kind of a bigger picture of all of those genes together. Anyway, so a mutation is just a change in an organism's DNA. There's many different types, the cell is especially vulnerable during replication. As you might imagine, when you guys have, some of you modeled replication, and the DNA has to split open, it's got that replication bubble, and there's a lot of action, a lot of stuff going on, so just more room for error. And one mutation type that's listed here is called a point mutation, which just straight up means you've got one nucleotide substituted for another. So let's say there's supposed to be an A there, instead there's a T. Or if you want to look at this example down here at the bottom, we're going to say the one on the left is normal. Here's your DNA, G, A, T, C, T, C, A, G, G, C, T, A. And then the RNA that would come off of that is what you would expect, G with C, A with U, T with A, C with G, and so on. And then the little codons, similar to the practice that you did in class, CUA, you could look that up on the chart and find out that stands for this amino acid. GAG represents this amino acid, and so on. Well, let's say there's a mutation in just one spot. So here, let's see, it was GATCTC, okay? Over here it's GATCTA. So this is the mutated base. So for whatever reason, this has come into play. Well, the RNA, therefore, that is transcribed off of that DNA is going to also be different. So instead of having a G over here, you're going to have a U. And this codon, GAU, is the code for ASP amino acid, not GLU. So you can see we've got a different amino acid. So if we have a whole bunch of amino acids that have things in them they're not supposed to have, they may not turn out the way they're supposed to. All right. So it's kind of like a typo. So again, many types can occur. Another kind of a common way to describe a mutation or a more common mutation is what we call a frame shift. And the name kind of helps explain it pretty well. In this case, you're going to insert or delete a nucleotide in the DNA sequence. Okay, so again, we're cruising along normal here like we did last time. Same string of DNA. Here's the RNA. Here's the protein. Okay, the central dogma, DNA, RNA, protein. Now let's say we have a frame shift mutation. There's supposed to be another G in here, but it's been deleted. So again, if you pop over here to the normal side, you got two G's next to each other. Well, one of them is missing here. So our reading frame, as it were, is going to change. So if I'm reading 3 at a time, GAT, CTC, now I'm going to read AGC instead of AGG. So my reading frame has shifted. All right, so in other words, I have a whole new set of codons. I mean, I was okay at first, GAT, CTC. That's the same as over here, GAT, CTC. But then I go AGG. Well, now there's no other G here, so I'm gonna go AGC. That's my new codon. And that winds up giving me, I still get that amino acid is the same as it turns out, but the next one is different. All right, so the frame has shifted because you're missing, or you have an extra one, either way. There's a good example in your book, by the way, that kind of does a sentence like the cat in the hat, and then like you take out a letter and it makes it just gibberish. Okay, now if you have a chromosomal mutation, that's a whole new ball game. We're saying a huge chunk of the chromosome. That means more than just one gene. That means a whole bunch of genes are being affected. A popular time for this to happen is during the process of crossing over. So if you think back to meiosis and stuff, but if you just remember, there's a time when the cell is duplicating its DNA. Chromosomes can actually sort of trade off bits and pieces from each other. So they're drawn here in purple and green so you can see the distinction. So somehow an entire segment of this chromosome, which is just a bunch of genes are in here. This is all super densely packed. Kind of like the clumps of DNA you saw in the lab today are just a big old, there's just a gazillion DNA molecules in there. Well, anyway, a whole chunk of this is being traded over to this purple one, and then this green one's getting the purple one back. So in other words, here's your result. This looks totally different. It's got a whole new set of genes on it. All right, so it's like an unequal crossing over. It wasn't supposed to work out that way. Another idea is called translocation, meaning we're not talking about two chromosomes that are what we call homologous, meaning like here's two copies of chromosome 1. Here's two copies of chromosome 17. Well, in this previous one, we could consider maybe this previous one is this is two copies of chromosome 1, and they've kind of traded segments with each other. We'll go back to this one. We've got two chromosome 1s, and then a totally different set of chromosomes, number 17. Remember, we have 23 pairs. We'll go over here. If they're translocated, it means, all right, a piece of number 1 didn't trade with the other number, its homologous pair, number 1. It traded with a completely different chromosome altogether. In this case, they just use the example of 1 switching up with 17. So that's translocation, a totally different spot. So mutations, again, like we said, may or may not affect phenotype. Chromosomal mutations have the largest effect on an organism. And here's some things that can happen when, moving on from chromosomal mutations into gene mutations, in the way in which they could change the phenotype, they could cause a premature stop codon. So if you're thinking about potential impact of mutations, premature stop codon, change the protein shape, or an incorrect amino acid, which would lead to the wrong protein. It could change gene regulation. Now, we didn't talk about gene regulation, so that was in section 8.6, so you can disregard that. Okay, and some of them, if you're looking at effects of mutation, there's a silent. Mutation can be what we call silent. There is no apparent effect. That's what it means to be silent. Maybe it happens in a region, what's called a non-coding region, or an area that's not used. it sounds strange, but not all the DNA is read. Kind of like if you're reading a book, maybe you don't read the introduction or something like that. So maybe that's where the mutation happens, and so no harm, no foul in that case. And maybe even though the mutation is there, the protein can still do its thing. Like it's not enough of a change to really make the protein dysfunctional. It's like, well, it is different. It's not exactly right. Kind of like maybe you have to walk with a limp, after you stub your toe, but you can still walk. So the protein's still able to do what it needs to do. It's just maybe not doing it quite the way it was doing it before. Okay, and then, just to make a point here about mutations in body cells. So they're just making a distinction here. I don't know if this really, it doesn't look like this fits on your notes very well. But I'm just going to say this real quick, is that, body cell is like your skin cell or something. So that's not going to get passed on to the next generation. So if you have a skin cell on you right now that mutates, your offspring wouldn't inherit that, okay? Whereas if it's in your egg or sperm, that could be harmful or perhaps beneficial in some cases to offspring. So you can pass on mutations that happen in what are called your gametes, your sex cells, what we would know as eggs and sperm. And then as a side note, and we'll talk about this in the future a lot, is that natural selection oftentimes will quote unquote remove mutant alleles from a population if they're less adaptive. So in other words, if a certain organism has a trait that doesn't give it any sort of advantage, sort of like, it's just something that's there. I want to think of a good example that's appropriate for the situation here, but we will talk a lot about it anyway, so I can just save it for that discussion on natural selection. But sometimes those will appear in a population. If they're not functional, they'll get taken out of the population. Okay, moving on. So these are the mutagens. These are the things that could cause the mutations. All right. Essentially, things in your environment. Okay, so UV rays, certain chemicals. There's so many, too many to name. And if you read the back of the package of something, you may read that it says has been known to cause cancer in laboratory rats or something. So we hear about so many things that cause cancer, and that sort of seems to change from time to time in the literature. This is safe, this is not safe. We know that that's sort of a fluctuating thing. It's like using your cell phone all the time, that causes cancer. Well, we really don't have evidence to show that that's true. So the popular opinion changes. We may not have been using cell phones long enough to even have enough data. But the point being, we know there are things out there that do cause cancer, that are definite cancer causers. UV rays are a definite, all right? Certain chemicals, like, you don't need to know them, but benzene, definitely going to cause you some cancers, or a mutation. And then sometimes the cancer drugs that you actually use to try to help people, they'll take advantage of this mutagenic property to kill cancer cells, all right? So we kind of use that to treat someone with cancer to actually, these are cells gone wild, so we can actually mutate those cells and kill them. So, and again, just to remind you, replication is when the cell is most vulnerable to mutations. All right, so just in summary, if you're looking back through your notes from the top, if you're looking on the left-hand side, gene mutations, those are mutations that affect a single gene. If you're looking on the right-hand side at the top, chromosomal mutations, those are mutations that affect a chromosome. All right, back on the left, you want to list the different mutation types, okay, for a single gene. On the right, you want to make sure you stick with things that are related specifically to chromosomes. All right, so just look back through the slides and make the distinction between the two. So it's kind of a compare and contrast. And then I think we hit mutations pretty good, so you should be in good shape. All right, good luck. We'll see you tomorrow.